Right now, as you listen to these words, your body is performing approximately 25 million billion chemical reactions every single second. In the time it takes you to draw one breath, enzymes in your cells have catalyzed more molecular transformations than there are stars in the observable universe. Yet, you feel none of this extraordinary activity. You sense no hint of the glucose molecules being systematically dismantled to fuel your thoughts. No awareness of the amino acids being assembled into the proteins that maintain your cellular architecture. No perception of the intricate molecular machinery that converts the oxygen in your lungs into the energy that powers your heartbeat. This hidden universe of chemistry is not separate from your experience of being alive. It is your experience of being alive. Every emotion you feel, every memory you form, every decision you make, emerges from the precise orchestration of molecules so small that millions could dance on the head of a pin. When you fall in love, you are witnessing the neurochemistry of dopamine and serotonin. When you feel energized after a meal, you are observing cellular respiration in action. When your muscles grow stronger through exercise, you are watching protein synthesis unfold in real time. Biochemistry is the fundamental language that translates between the world of atoms and the world of life. It explains how simple carbon, hydrogen, oxygen, and nitrogen become the complexity of consciousness. It reveals why certain foods nourish you while others harm you, how your body repairs itself while you sleep, and why aging occurs at the molecular level. I'm curious where you're listening from tonight and what time it is in your corner of the world. However you've found your way here, I'm delighted you've joined me for this exploration into the molecular foundations of existence itself. Let's dive into the chemistry of life. To understand biochemistry, we must first understand the elemental foundation upon which all biological processes rest. Life, in all its magnificent complexity, emerges from the interactions of just a handful of chemical elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur form the core constituency of living matter, though they are joined by essential traces of calcium, potassium, sodium, chlorine, magnesium and iron. These elements are not randomly distributed throughout biological systems but are organized according to predictable chemical principles that have remained constant since life first emerged on Earth. Carbon stands as the undisputed backbone of biological chemistry. Its unique ability to form 4 stable covalent bonds creates virtually unlimited possibilities for molecular architecture. Unlike other elements, carbon can bond with itself to form long chains, branched networks, and ring structures, providing the scaffolding for the complex molecules that make life possible. This versatility explains why carbon-based chemistry, rather than silicon or any other element, became the foundation of terrestrial biology. The bonds that hold these atoms together determine the properties and behaviors of biological molecules. Covalent bonds, formed when atoms share electrons, create the stable frameworks of proteins, carbohydrates, lipids, and nucleic acids. These bonds are strong enough to maintain molecular integrity, yet can be broken and reformed through enzymatic processes, allowing for the dynamic chemistry that characterizes living systems. Ionic bonds, formed when electrons are transferred from one atom to another, play crucial roles in protein structure and cellular signaling. The attraction between positively and negatively charged ions helps stabilize the three-dimensional shapes of proteins and enables the rapid transmission of electrical signals in neurons. Sodium and potassium ions, for instance, create the electrical gradients that power nerve impulses and muscle contractions. Perhaps most critically for life, hydrogen bonds provide the subtle yet essential forces that allow biological molecules to interact with precision. These relatively weak attractions between hydrogen atoms and electronegative atoms like oxygen or nitrogen enable DNA strands to recognize their complementary partners, allow proteins to fold into their functional shapes, and give water its extraordinary properties as a biological solvent. Water deserves special attention as the universal medium of life. Its unique molecular structure, with 2 hydrogen atoms bonded to 1 oxygen atom at an angle of 104 degrees, creates a polar molecule with distinct positive and negative regions. This polarity allows water to dissolve a remarkable variety of biological molecules, earning it the title of universal solvent. More than mere passive medium, water participates actively in biological processes, from the hydrolysis reactions that break down nutrients to the photosynthetic reactions that capture solar energy. The hydrogen bonding between water molecules creates properties essential for life. Water remains liquid across the temperature range where biological processes occur. It has a high specific heat that buffers organisms against temperature fluctuations, and it becomes less dense when frozen, allowing ice to float and protecting aquatic life during winter months. Without these unique properties, Earth's biosphere as we know it could not exist. Biological systems must also maintain precise chemical environments through careful regulation of acidity and alkalinity. The pH scale, ranging from 0 to 14, measures the concentration of hydrogen ions in solution. Pure water with equal concentrations of hydrogen and hydroxide ions registers a neutral pH of 7. Most biological processes occur within a narrow pH range around neutrality, typically between 6.8 and 7.4. Living organisms achieve this pH stability through sophisticated buffer systems that resist changes in acidity. The bicarbonate buffer system in blood, for example, maintains plasma pH within the narrow range, essential for enzyme function and cellular metabolism. When carbon dioxide from cellular respiration dissolves in blood, it forms carbonic acid, which could dangerously lower blood pH. The bicarbonate buffer system neutralizes this acid, preventing the pH fluctuations that would disrupt vital physiological processes. The chemistry of life is further organized around functional groups, specific arrangements of atoms that confer particular chemical properties to organic molecules. Hydroxyl groups make molecules polar and able to torque hydrogen bonds. Carboxyl groups act as acids, donating hydrogen ions in solution. Amino groups act as bases, accepting hydrogen ions. Phosphate groups carry negative charges and can link molecules together or store energy in their bonds. These functional groups act like chemical tools, enabling molecules to interact in predictable ways that drive the processes of life. Understanding these fundamental chemical principles illuminates why biochemistry follows predictable patterns, despite its apparent complexity. The same forces that determine how atoms bond in a laboratory beaker also govern how enzymes recognize their substrates, how hormones bind to their receptors, and how genetic information flows from DNA to proteins. Chemistry becomes biology through the elegant organization of these basic principles into increasingly complex systems, each level building upon the foundation of atomic interactions that obey universal physical laws. In the hierarchy of biological molecules, carbohydrates serve as both the immediate fuel that powers cellular processes and the structural framework that gives shape to living organisms. These versatile compounds, composed of carbon, hydrogen, and oxygen in roughly equal proportions, represent the most direct link between the energy of sunlight and the energy demands of life itself. The simplest carbohydrates are monosaccharides, single sugar units that cannot be broken down into smaller carbohydrate components. Glucose stands as the most important of these molecular building blocks, serving as the primary energy currency for virtually all living cells. Its 6-carbon structure, with the molecular formula C6H12O6, represents the perfect balance of stability and reactivity that makes it ideal for biological energy storage and transfer. When you consume a meal containing starches or sugars, digestive enzymes rapidly convert complex carbohydrates into glucose, which then enters your bloodstream and travels to cells throughout your body. This glucose serves as the immediate fuel for cellular respiration, the process by which cells extract usable energy from organic molecules. The brain alone consumes approximately 20% of your body's total glucose supply, despite representing only 2% of your body weight, highlighting the critical importance of this simple sugar for nervous system function. Other monosaccharides play specialized roles in biological systems. Fructose, found naturally in fruits and honey, follows a slightly different metabolic pathway than glucose, but ultimately contributes to the same energy-producing processes. Galactose, derived from milk sugar, requires specific enzymes for its metabolism, and genetic deficiencies in these enzymes can lead to serious metabolic disorders. Ribose, the 5-carbon sugar, forms the backbone of RNA and plays essential roles in energy transfer through molecules like ATP. When two monosaccharides link together through glycosidic bonds, they form disaccharides that serve both nutritional and functional purposes. Sucrose, common table sugar derived from sugarcane and sugar beets, consists of glucose and fructose joined together. Lactose, the sugar found in mammalian milk, combines glucose and galactose to provide energy for developing offspring. Maltose, formed when starch breaks down during digestion, represents an intermediate step in the conversion of complex carbohydrates to simple sugars. The real structural and storage power of carbohydrates emerges when hundreds or thousands of monosaccharide units link together to form polysaccharides. These complex carbohydrates demonstrate the remarkable versatility of sugar chemistry, creating molecules that range from energy storage depots to rigid structural materials. Glycogen represents the animal kingdom's solution to energy storage. This highly branched polymer of glucose can be rapidly assembled when energy is abundant and quickly dismantled when cellular fuel runs low. Your liver stores approximately 100 grams of glycogen, while your muscles contain roughly 400 grams distributed throughout skeletal muscle tissue. During periods of fasting or intense exercise, enzymes break down glycogen through a process called glycogenolysis, releasing glucose units that can immediately enter energy-producing pathways. Plants store energy in the form of starch, a polymer of glucose that exists in two forms. Amylose consists of long, unbranched chains of glucose units, while amylopectin features extensive branching that allows for more compact storage and rapid access to glucose when needed. When you consume foods like potatoes, rice, or bread, digestive enzymes systematically break down these starch molecules, releasing glucose for absorption into your bloodstream. The process by which cells extract energy from glucose involves a carefully orchestrated series of chemical reactions collectively known as glycolysis. This ancient metabolic pathway, which occurs in the cytoplasm of virtually all living cells, breaks down one molecule of glucose into two molecules of pyruvate, while generating a net gain of two ATP molecules and reducing equivalents that can be used for further energy production. Glycolysis begins when glucose enters a cell and becomes trapped by phosphorylation, preventing it from leaving the cell and committing it to metabolic processing. Through a series of 10 enzymatic steps, the 6-carbon glucose molecule is systematically rearranged and ultimately cleaved into two 3-carbon pyruvate molecules. This process requires an initial investment of 2 ATP molecules, but produces 4 ATP molecules, resulting in a net energy gain that can immediately support cellular processes. When glucose supplies run low, your body activates gluconeogenesis, the metabolic pathway that synthesizes new glucose from non-carbohydrate precursors. This process essentially runs glycolysis in reverse, using amino acids from protein breakdown, lactate from muscle metabolism, or glycerol from fat breakdown to construct glucose molecules. Gluconeogenesis primarily occurs in the liver and kidneys, ensuring that glucose-dependent tissues like the brain maintain adequate fuel supplies even during periods of carbohydrate restriction. The pentose phosphate pathway provides an alternative route for glucose metabolism that serves specialized cellular needs. Rather than focusing primarily on energy production, this pathway generates NADPH, a reducing agent essential for biosynthetic reactions and antioxidant defense systems. The pathway also produces ribose 5-phosphate, which serves as the backbone for nucleotide synthesis. Red blood cells rely heavily on the pentose phosphate pathway to maintain the reducing environment necessary for protecting hemoglobin from oxidative damage. Blood sugar regulation represents one of the most tightly controlled aspects of human physiology, involving multiple organ systems working in concert to maintain glucose concentrations within narrow limits. When blood glucose levels rise after a meal, beta cells in the pancreatic islets of Langerhans respond by secreting insulin, a hormone that promotes glucose uptake by muscle and fat cells while stimulating glycogen synthesis in the liver. Conversely, when blood glucose levels fall, alpha cells in the pancreas release glucagon, which stimulates glycogen breakdown in the liver and promotes gluconeogenesis. This hormonal control system maintains blood glucose concentrations between approximately 70 and 100 milligrams per deciliter under normal conditions, preventing both the energy deficits associated with hypoglycemia and the tissue damage that can result from chronic hyperglycemia. The intricate relationship between carbohydrate metabolism and overall health becomes apparent in conditions like diabetes mellitus, where disruptions in insulin signaling lead to persistent hyperglycemia and wide-ranging metabolic consequences. Understanding how carbohydrates function at the molecular level provides insight into both the fundamental processes that sustain life and the therapeutic approaches used to treat metabolic disorders that affect millions of people worldwide. While carbohydrates provide the immediate energy for cellular processes, lipids serve as the body's long-term energy storage specialists, structural architects, and molecular messengers. These diverse compounds, unified by their hydrophobic nature and solubility in non-polar solvents, demonstrate remarkable versatility in biological systems, From the membranes that define cellular boundaries to the hormones that coordinate physiological responses across vast distances, the foundation of lipid chemistry rests upon fatty acids, long hydrocarbon chains terminated by a carboxyl group that gives them their acidic properties. These molecules vary primarily in their length and degree of saturation, characteristics that profoundly influence their physical properties and biological functions. Saturated fatty acids contain no double bonds between carbon atoms, allowing them to pack tightly together and remain solid at room temperature. Palmitic acid and stearic acid, common saturated fatty acids found in animal fats, exemplify this molecular architecture. Unsaturated fatty acids contain one or more double bonds that introduce kinks into their otherwise straight chains, preventing tight packing and keeping them liquid at physiological temperatures. Oleic acid, the primary component of olive oil, contains a single double bond and remains fluid at body temperature. Polyunsaturated fatty acids like linoleic acid and alpha-linolenic acid contain multiple double bonds and play essential roles in membrane structure and signaling molecule synthesis. The body's most efficient energy storage system relies on triglycerides, molecules formed when three fatty acids bond to a glycerol backbone through ester linkages. This molecular arrangement allows enormous amounts of energy to be stored in relatively small volumes. A single gram of fat provides approximately 9 calories of energy, more than twice the energy density of carbohydrates or proteins. Adipose tissue, composed primarily of cells filled with triglycerides, serves as the body's energy bank, capable of sustaining life for weeks or even months during periods of food scarcity. When energy demands its immediate glucose supplies, hormone-sensitive lipase breaks down triglycerides through lipolysis, releasing fatty acids into the bloodstream where they can travel to energy-demanding tissues. These fatty acids undergo beta-oxidation, a process that systematically removes 2 carbon units from the fatty acid chain, generating acetyl-CoA molecules that enter the citric acid cycle for complete oxidation to carbon dioxide and water. The efficiency of fat metabolism becomes apparent when considering that a single palmitic acid molecule yields 129 ATP molecules through complete oxidation, compared to the 38 ATP molecules generated from one glucose molecule. This remarkable energy yield explains why migrating animals and hibernating mammals rely heavily on fat stores to fuel their extended periods of high energy demand or metabolic suppression. Phospholipids represent a specialized class of lipids that form the structural foundation of all biological membranes. These amphipathic molecules contain both hydrophilic and hydrophobic regions, allowing them to spontaneously organize into bilayer structures that separate aqueous compartments while maintaining selective permeability. Phosphatidylcholine and phosphatidylserine, abundant in cell membranes, demonstrate how subtle variations in polar head groups can influence membrane properties and cellular functions. The phosphosolipid bilayer creates a barrier that is impermeable to most polar molecules, while allowing nonpolar substances to pass freely. This selective permeability enables cells to maintain distinct internal environments while facilitating the controlled exchange of materials with their surroundings. Membrane proteins embedded within the phospholipid matrix serve as gates, channels, and pumps that regulate molecular traffic across cellular boundaries. Cholesterol occupies a unique position in lipid biochemistry, serving simultaneously as a membrane component and a precursor for numerous signaling molecules. Despite its reputation in popular culture, cholesterol plays essential roles in maintaining membrane fluidity and serves as the starting material for steroid hormone synthesis. The rigid steroid backbone of cholesterol intercalates between phospholipid molecules, modulating membrane fluidity and ensuring optimal conditions for membrane protein function. The synthesis of cholesterol represents one of the most complex biosynthetic pathways in human metabolism, requiring more than 30 enzymatic steps to convert acetyl-CoA into the final steroid product. This pathway is tightly regulated through feedback inhibition, with cholesterol itself suppressing the activity of key regulatory enzymes when cellular cholesterol levels become adequate. Steroid hormones derived from cholesterol coordinate numerous physiological processes through their ability to regulate gene expression. Testosterone and estradiol control reproductive development and function. Cortisol manages stress responses and glucose metabolism, and aldosterone regulates sodium and potassium balance in the kidneys. These hormones demonstrate the remarkable transformation of a simple lipid molecule into powerful regulatory signals that influence cellular behavior throughout the body. The process of fatty acid synthesis occurs primarily in the liver and adipose tissue when carbohydrate intake exceeds immediate energy needs. Acetyl-CoA carboxylase catalyzes the rate-limiting step of this pathway, converting acetyl-CoA to malonyl-CoA, which then serves as the building block for fatty acid chain elongation. Fatty acid synthase, a large multi-enzyme complex, systematically adds 2 carbon units to the growing fatty acid chain until palmitic acid is produced. Essential fatty acids represent a special category of lipids that the human body cannot synthesize, but requires for optimal health. Linoleic acid and alpha-linolenic acid serve as precursors for longer-chain polyunsaturated fatty acids that play crucial roles in inflammation regulation, brain development, and cardiovascular health. Arachidonic acid, derived from linoleic acid, serves as the precursor for prostaglandins, leukotrienes, and other signaling molecules that mediate inflammatory responses and pain perception. The omega-3 fatty acids, including eicosapentaenoic acid and docosahexaenoic acid, demonstrate the profound influence that dietary lipids can have on human health. These fatty acids incorporate into cell membranes, particularly in the brain and retina, where they influence membrane fluidity and support optimal neuronal function. They also serve as precursors for specialized pro-resolving mediators that help resolve inflammatory responses and promote tissue healing. Lipid metabolism disorders illustrate the critical importance of proper fat processing in human health. Familial hypercholesterolemia results from defective cholesterol receptors, leading to elevated blood cholesterol levels and increased cardiovascular disease risk. Fatty acid oxidation disorders can cause severe hypoglycemia and metabolic crises when the body cannot efficiently utilize stored fat for energy production. The intricate regulation of lipid metabolism involves complex interactions between hormonal signals, nutritional status, and genetic factors, demonstrating how these seemingly simple molecules participate in some of the most sophisticated regulatory networks in human physiology. Proteins represent the most structurally diverse and functionally versatile class of biological molecules, serving as the molecular machines that carry out virtually every process essential to life. From the enzymes that catalyze biochemical reactions to the structural components that give cells their shape, proteins demonstrate how a relatively simple chemical alphabet can generate unlimited functional complexity through precise three-dimensional organization. The foundation of protein structure rests upon 20 standard amino acids, each containing a central carbon atom bonded to an amino group, a carboxyl group, a hydrogen atom, and a distinctive side chain that determines the amino acid's unique properties. These side chains vary dramatically in their chemical characteristics, ranging from the simple hydrogen atom of glycine to the complex aromatic rings of tryptophan and phenylalanine. Some side chains are hydrophobic, preferring to avoid contact with water, while others are hydrophilic, readily forming hydrogen bonds or ionic interactions with surrounding molecules. The chemical diversity of amino acid side chains provides the basis for protein function. Serine and threonine contain hydroxyl groups that can participate in chemical reactions or form hydrogen bonds. Cysteine contains a sulfur atom that can form disulfide bonds with other cysteine residues, creating crosslinks that stabilize protein structure. Histine can accept or donate protons, making it valuable in enzyme active sites where pH changes are important for catalytic activity. Protein structure unfolds across 4 distinct levels of organization, each building upon the previous level to create molecules of extraordinary complexity. Primary structure refers to the linear sequence of amino acids connected by peptide bonds, formed when the carboxyl group of one amino acid condenses with the amino group of the next, releasing a water molecule in the process. This sequence, determined by genetic information, dictates all higher levels of protein organization through the chemical properties of the constituent amino acids. Secondary structure emerges from regular repeating arrangements of the protein backbone, stabilized by hydrogen bonds between backbone atoms. Alpha helices represent one common secondary structure, where the protein backbone spirals in a right-handed helix with hydrogen bonds forming between carbonyl oxygen atoms and amino hydrogen atoms, 4 residues away. Beta sheets represent another fundamental secondary structure, where extended protein chains lie adjacent to one another, forming hydrogen bonds between backbone atoms of neighboring strands. Tertiary structure describes the overall 3-dimensional arrangement of a single protein chain, determined by interactions between amino acid side chains, separated by large distances in the primary sequence. Hydrophobic side chains tend to cluster together in the protein interior, excluding water and creating a stable core, Charged side chains often locate on the protein surface, where they can interact favorably with the aqueous environment. Disulfide bonds between cysteine residues can cross-link distant parts of the protein chain, providing additional structural stability. Quaternary structure applies to proteins composed of multiple polypeptide chains, called subunits, that associate to form a functional complex. Hemoglobin exemplifies quaternary structure, consisting of 4 subunits that work together to transport oxygen through the bloodstream. The interactions between subunits can be regulated, allowing quaternary structure changes to modulate protein function in response to cellular conditions. Enzymes represent the most sophisticated class of proteins functioning as biological catalysts that accelerate chemical reactions by lowering the activation energy required for bond breaking and formation. The active site of an enzyme creates a specialized microenvironment where substrates bind with precise geometric and chemical complementarity. This binding brings reactive groups into optimal positions for catalysis, while stabilizing the transition state of the reaction, dramatically increasing the reaction rate. The lock and key model initially described enzyme specificity, suggesting that substrates fit into enzyme active sites like keys fitting into locks. The induced fit model provides a more accurate description, recognizing that both enzyme and substrate undergo conformational changes upon binding, optimizing the interaction for catalysis. This flexibility allows enzymes to achieve remarkable specificity while maintaining the dynamic properties necessary for efficient catalysis. Enzyme kinetics follows predictable mathematical relationships that describe how reaction rates depend on substrate concentrations and enzyme properties. The Michaelis-Menten equation characterizes this relationship, defining parameters that quantify enzyme efficiency and substrate affinity. Understanding these kinetic principles enables researchers to design drugs that modulate enzyme activity and to engineer enzymes with improved properties for industrial applications. The process of protein folding represents one of the most fascinating problems in molecular biology, as a linear sequence of amino acids spontaneously adopts a specific three-dimensional structure necessary for function. This folding process occurs through a complex energy landscape, where the protein samples numerous conformations before settling into its native state. Molecular chaperones assist this process by preventing inappropriate aggregation and helping proteins navigate folding pathways efficiently. Protein misfolding can have devastating consequences for human health, as demonstrated by diseases like Alzheimer's and Parkinson's, where specific proteins aggregate into toxic structures that damage cells. The prion diseases represent an extreme example of protein misfolding, where abnormally folded proteins can induce normal proteins to adopt the misfolded conformation, creating a self-propagating pathological process. Protein synthesis occurs through the coordinated action of ribosomes, transfer RNAs, and numerous accessory factors that translate the genetic code into polypeptide sequences. This process begins when messenger RNA binds to a ribosome, positioning the start codon in the appropriate site for translation initiation. Transfer RNAs deliver amino acids to the ribosome in the order specified by the mRNA sequence, and peptide bonds form between successive amino acids as the ribosome moves along the mRNA. The fidelity of protein synthesis depends on the accurate recognition of codons by transfer RNAs and the proofreading activities of aminoacyl tRNA synthetases, enzymes that attach amino acids to their corresponding transfer RNAs. Errors in protein synthesis can produce defective proteins with altered functions, highlighting the importance of quality control mechanisms in maintaining cellular health. Protein degradation represents an equally important aspect of protein metabolism, as damaged or unnecessary proteins must be removed to maintain cellular function. The ubiquitin proteasome system tags proteins for destruction by attaching ubiquitin molecules to specific lysine residues, creating a signal that directs the protein to the proteasome for degradation. This system allows cells to rapidly adjust protein levels in response to changing conditions, and to eliminate potentially harmful misfolded proteins. Structural proteins provide the architectural framework that gives cells and tissues their mechanical properties. Collagen, the most abundant protein in mammals, forms rope-like structures that provide tensile strength to skin, tendons, and blood vessels. The unique amino acid composition of collagen, with frequent glycine residues and abundant hydroxyproline, allows 3 polypeptide chains to wrap around each other in a stable triple helix that resists stretching forces. Keratin represents another important structural protein, forming intermediate filaments that provide mechanical strength to epithelial cells. The hierarchical organization of keratin, from individual molecules to complex fiber networks, demonstrates how protein structure spans multiple length scales to create materials with specific mechanical properties. Transport proteins facilitate the movement of molecules across biological membranes and through body fluids, overcoming the selective permeability barriers that separate cellular compartments. Hemoglobin exemplifies transport protein function, using conformational changes triggered by oxygen binding to enhance oxygen delivery efficiency. The cooperative binding of oxygen to hemoglobin creates a sigmoidal binding curve that optimizes oxygen pickup in the lungs and release in metabolically active tissues. Nucleic acids represent the ultimate information storage and processing system in living organisms, encoding the instructions that determine every aspect of cellular structure and function. These remarkable polymers, composed of nucleotide building blocks, arranged in precise sequences, serve as both the repository of hereditary information and the dynamic machinery that translates genetic instructions into functional proteins. Each nucleotide consists of 3 essential components that work together to create the foundation of genetic information storage. The nitrogenous base provides the information carrying capacity through its specific chemical structure and hydrogen bonding properties. The 5-carbon sugar forms the backbone structure that connects nucleotides into long chains. The phosphate group creates the acidic character that gives nucleic acids their name and provides the energy for certain cellular processes. The nitrogenous bases fall into 2 distinct chemical categories that determine their pairing properties and biological functions. Purines, including adenine and guanine, contain double ring structures that provide stability and specific hydrogen bonding patterns. Pyrimidines, including cytosine, thymine, and uracil, feature single-ring structures that complement the purines through precise geometric relationships. This chemical complementarity forms the basis for the specific base pairing rules that ensure accurate information storage and transfer. DNA achieves its remarkable stability and information storage capacity through its iconic double helix structure, first elucidated by Watson and Crick. Through careful analysis of X-ray crystallography data, 2 anti-parallel strands of DNA wind around each other in a right-handed spiral, with the sugar-phosphate backbones forming the outer framework and the paired bases, creating the internal structure. Adenine always pairs with thymine through 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds, creating the complementary base pairing that allows each strand to serve as a template for the other. The double helix structure provides multiple layers of protection for genetic information. The paired bases, stacked in the interior of the helix, are shielded from chemical damage by the surrounding sugar-phosphate framework. The complementary nature of the 2 strands means that damage to one strand can be repaired using the intact strand as a template. The helical structure itself provides mechanical stability while allowing controlled access to the genetic information when needed for replication or transcription. RNA molecules demonstrate remarkable structural and functional diversity despite sharing basic chemical similarities with DNA. The presence of ribose rather than deoxyribose in the sugar backbone makes RNA less chemically stable than DNA, but more structurally flexible, allowing RNA molecules to adopt complex 3-dimensional shapes necessary for their diverse functions. The substitution of uracil for thymine in RNA enables different base pairing patterns that contribute to RNA's catalytic and regulatory capabilities. Messenger RNA serves as the intermediary between genetic information stored in DNA and the protein synthesis machinery of the cell. These molecules carry copies of genetic instructions from the nucleus to the ribosomes, where the information is translated into protein sequences. The structure of mRNA includes coding sequences that specify amino acid sequences, as well as regulatory regions that control when and how efficiently the message is translated. Transfer RNA molecules function as adapter molecules that interpret the genetic code by bringing specific amino acids to the ribosome during protein synthesis. Each transfer RNA has a distinctive cloverleaf secondary structure that folds into a compact L-shaped 3-dimensional structure. The anticodon region of transfer RNA recognizes specific codons on messenger RNA through complementary base pairing, while the amino acid attachment site ensures that the correct amino acid is incorporated into the growing protein chain. Ribosomal RNA represents the catalytic component of ribosomes, the cellular machines responsible for protein synthesis. These RNA molecules not only provide structural framework for ribosome assembly, but also catalyze the formation of peptide bonds between amino acids. The discovery that RNA, rather than protein, catalyzes this fundamental biological process revolutionized our understanding of the relationship between nucleic acids and proteins in cellular function. DNA replication represents one of the most precisely controlled processes in biology, ensuring that genetic information passes accurately from one generation to the next. This process begins when specialized enzymes unwind the double helix, creating replication forks where the two strands separate and become accessible to the replication machinery. DNA polymerases synthesize new DNA strands by adding nucleotides that are complementary to the template strands, following the base pairing rules that ensure accurate copying. The semi-conservative nature of DNA replication means that each new double helix contains one original strand and one newly synthesized strand. This mechanism provides a built-in quality control system, as the original strand serves as a permanent template against which the accuracy of the new strand can be checked. Proofreading activities of DNA polymerases and additional repair mechanisms ensure that replication errors occur less than once in every billion nucleotides incorporated. Transcription initiates gene expression by creating RNA copies of specific DNA sequences. RNA polymerase enzymes bind to promoter sequences that mark the beginning of genes and synthesize RNA molecules that are complementary to one strand of the DNA template. Unlike DNA replication, which copies entire chromosomes, transcription is highly selective, producing RNA copies of only those genes that are needed under specific cellular conditions. The process of transcription involves multiple levels of regulation that allow cells to control gene expression with remarkable precision. Transcription factors bind to specific DNA sequences and either promote or inhibit RNA polymerase activity. Enhancer and silencer sequences can influence transcription from great distances, creating complex regulatory networks that coordinate gene expression across entire cellular programs. Translation converts the nucleotide sequence information in messenger RNA into the amino acid sequences of proteins through the universal genetic code. This code consists of 64 3-nucleotide codons that specify the 20 standard amino acids, plus stop signals that terminate protein synthesis. The redundancy built into the genetic code provides protection against mutations, as many amino acids are specified by multiple codons, allowing some sequence changes to occur without altering the protein product. The ribosome coordinates the complex process of translation by bringing together messenger RNA, transfer RNAs, and various protein factors in the precise arrangements necessary for accurate protein synthesis. The ribosome moves along the messenger RNA in 3 nucleotide steps, ensuring that the genetic code is read in the correct frame and that amino acids are incorporated in the order specified by the gene sequence. DNA repair mechanisms demonstrate the critical importance of maintaining genetic integrity in living systems. Multiple pathways exist to correct different types of DNA damage, from simple base modifications to complex double-strand breaks. Mismatch repair systems recognize and correct errors that escape proofreading during replication. Base excision repair remove damaged or inappropriate bases and replaces them with correct nucleotides. Nucleotide excision repair handles bulky DNA lesions that distort the double helix structure. The sophisticated nature of DNA repair systems reflects the constant challenge that living organisms face in preserving genetic information against chemical damage, radiation, and replication errors. Defects in DNA repair pathways can lead to increased mutation rates and predisposition to cancer, highlighting the essential role these systems play in maintaining cellular and organismal health. Enzymes represent the most sophisticated catalytic systems known to science, orchestrating virtually every chemical reaction that occurs within living organisms. These remarkable protein molecules accelerate biochemical reactions by factors of millions or even billions, transforming processes that would otherwise require extreme temperatures or harsh chemical conditions into gentle reactions that proceed efficiently at body temperature and physiological pH. The fundamental principle underlying enzyme function involves the lowering of activation energy, the energy barrier that must be overcome for a chemical reaction to proceed. Without enzymes, most biochemical reactions would occur so slowly that life as we know it would be impossible. A simple reaction like the hydrolysis of sucrose might take years to complete under physiological conditions, but the enzyme sucrase catalyzes the same reaction in milliseconds. Enzymes achieve this catalytic acceleration through the creation of specialized microenvironments within their active sites. These precisely sculpted molecular cavities provide optimal conditions for chemical transformation by positioning reactive groups in perfect geometric arrangements, stabilizing transition states, and excluding water molecules that might interfere with the reaction. The active site represents the culmination of millions of years of evolutionary refinement, creating molecular machines of extraordinary precision and efficiency. The structure of enzyme active sites reveals the intimate relationship between protein architecture and catalytic function. Amino acid residues from distant parts of the protein chain converge to create the active site through precise three-dimensional folding. Some residues bind substrate molecules through specific hydrogen bonds or electrostatic interactions, while others participate directly in catalysis by donating or accepting protons, forming temporary covalent bonds, or stabilizing charged intermediates. The induced fit model describes how enzymes achieve optimal catalytic efficiency through dynamic structural changes. When substrate molecules approach the active site, both enzyme and substrate undergo conformational adjustments that optimize their interaction. This mutual adaptation ensures maximum complementarity between enzyme and substrate at the transition state, the fleeting intermediate structure that represents the highest energy point along the reaction pathway. Enzyme kinetics provides quantitative insight into catalytic mechanisms and regulatory properties through mathematical analysis of reaction rates under different conditions. The Michaelis-Menten equation describes the relationship between substrate concentration and reaction velocity, defining key parameters that characterize enzyme performance. The Michaelis constant represents the substrate concentration at which the enzyme operates at half its maximum velocity, providing a measure of enzyme affinity for its substrate. Maximum velocity reflects the catalytic efficiency of the enzyme when all active sites are occupied by substrate molecules. The ratio of these parameters, known as catalytic efficiency, provides a single number that summarizes overall enzyme performance and allows comparison between different enzymes or between wild-type and mutant versions of the same enzyme. Allosteric regulation represents a sophisticated mechanism by which enzyme activity responds to cellular conditions through binding of regulatory molecules at sites distinct from the active site. These allosteric effectors can enhance or inhibit enzyme activity by inducing conformational changes that either optimize or disrupt the active site structure. This mechanism allows ENs to serve as molecular switches that integrate multiple cellular signals. The cooperative binding exhibited by many allosteric enzymes creates sigmoidal kinetic curves that enable sensitive responses to small changes in substrate or effector concentrations. This cooperativity allows metabolic pathways to switch rapidly between different states, ensuring efficient adaptation to changing cellular needs. Cofactors and coenzymes extend the catalytic capabilities of enzymes beyond what can be achieved with amino acid side chains alone. These small molecules participate directly in catalytic processes, often serving as carriers for electrons, protons, or chemical groups that are transferred during the reaction. Many cofactors are derived from vitamins, explaining why these nutrients are essential for human health despite being required in only small quantities. Metal ion cofactors like zinc, iron, and magnesium provide unique chemical properties that are essential for specific types of reactions. Zinc ions facilitate hydrolysis reactions through their ability to polarize water molecules and stabilize negative charges. Iron-containing cofactors participate in electron transfer reactions and oxygen activation processes. Magnesium ions help position phosphate groups for transfer reactions and neutralize negative charges that would otherwise destabilize reaction intermediates. Coenzymes like NAD, F8, and coenzyme A function as recyclable carriers that shuttle chemical groups between different enzymes. NAD serves as an electron carrier in oxidation-reduction reactions, accepting electrons from one enzyme and delivering them to another. Coenzyme A activates carboxylic acids by forming high-energy thioester bonds that drive subsequent synthetic reaction. Enzyme inhibition provides crucial mechanisms for regulating metabolic pathways and represents the basis for many therapeutic drugs. Competitive inhibitors resemble natural substrates and compete for binding to the active site, reducing enzyme activity in a manner that can be overcome by increasing substrate concentration. Non-competitive inhibitors bind to allosteric sites and reduce enzyme activity regardless of substrate concentration, providing a different mode of regulation. Feedback inhibition represents an elegant regulatory mechanism, where the end product of a metabolic pathway inhibits the first enzyme in the pathway, preventing overproduction of the final product. This negative feedback creates self-regulating systems that maintain metabolic homeostasis without requiring external control mechanisms, Regulatory enzymes occupy key positions in metabolic pathways where they control the flux of metabolites through alternative routes. These enzymes often catalyze irreversible reactions that serve as committed steps, ensuring that metabolites proceed through specific pathways once the regulatory enzyme is activated. The careful positioning of regulatory enzymes allows cells to coordinate complex metabolic networks with remarkable precision. The practical applications of enzyme technology demonstrate the versatility and power of these biological catalysts. Industrial enzymes produce everything from high-fructose corn syrup to biodegradable detergents, often replacing harsh chemical processes with environmentally friendly alternatives. The specificity of enzymes allows them to perform selective transformations that would be difficult or impossible to achieve through traditional chemical synthesis. Medical applications of enzymes range from diagnostic assays that detect specific diseases to therapeutic enzymes that treat genetic disorders. Enzyme replacement therapy provides functional enzymes to patients who lack specific enzymes due to genetic mutations, offering treatment for previously incurable conditions. The development of modified enzymes with improved stability or altered specificity continues to expand the therapeutic potential of enzyme-based medicines. The study of enzyme mechanisms continues to reveal new principles of biological catalysis and inspire the design of artificial catalysts. Understanding how enzymes achieve their remarkable efficiency and specificity provides blueprints for creating synthetic catalysts that could revolutionize chemical manufacturing and environmental remediation. The marriage of biochemical knowledge with engineering principles promises to unlock new applications for these molecular machines that have perfected the art of chemical transformation over billions of years of evolution. Cellular respiration represents the most fundamental energy conversion process in living organisms, transforming the chemical energy stored in organic molecules into the universal energy currency that powers every aspect of cellular function. This sophisticated biochemical pathway demonstrates how cells have evolved elegant solutions to extract maximum energy from nutrients while maintaining the precise control necessary for life. The overall process of cellular respiration can be summarized as the controlled oxidation of glucose in the presence of oxygen, producing carbon dioxide, water, and approximately 38 molecules of adenosine triphosphate. This equation, however, belies the remarkable complexity of the molecular machinery that achieves this energy transformation through three interconnected stages, each optimized for specific aspects of energy extraction and conservation. Glycolysis initiates cellular respiration in the cytoplasm, where glucose undergoes a carefully orchestrated 10-step transformation into two molecules of pyruvate. This ancient metabolic pathway operates in virtually all living cells and can function in the absence of oxygen, suggesting its evolutionary origins in the anaerobic environment of early Earth. The process begins with an energy investment phase where two ATP molecules are consumed to phosphorylate glucose and its derivatives, trapping these molecules within the cell and destabilizing them for subsequent breakdown. The energy payoff phase of glycolysis more than compensates for this initial investment, producing 4 ATP molecules and 2 molecules of NADH through substrate-level phosphorylation and coupled oxidation-reduction reactions. The net gain of 2 ATP molecules provides immediate energy for cellular processes, while the NADH molecules serve as electron carriers that can generate additional ATP through subsequent oxidative processes. The conversion of glucose to pyruvate occurs through a series of carefully balanced chemical transformations that illustrate fundamental principles of biochemical regulation. Key regulatory enzymes like phosphofructokinase respond to cellular energy status through allosteric interactions with ATP, ADP, and other metabolic intermediates. When cellular energy levels are high, ATP binding inhibits phosphofructokinase activity, slowing glycolysis and conserving glucose for other purposes. Conversely, when energy demands increase, ADP and AMP binding stimulates the enzyme, accelerating glucose breakdown to meet cellular needs. The fate of pyruvate depends critically on oxygen availability and cellular energy demands. Under aerobic conditions, pyruvate enters the mitochondria through specific transport proteins and undergoes oxidative decarboxylation to form acetyl-CoA, the universal fuel molecule that feeds into the citric acid cycle. This reaction, catalyzed by the pyruvate dehydrogenase complex, represents a critical regulatory point, that commits pyruvate to complete oxidation while generating additional NADH and carbon dioxide. The citric acid cycle, also known as the Krebs cycle or tricarboxylic acid cycle, represents the central hub of cellular metabolism, where the complete oxidation of acetyl-CoA occurs through a cyclical series of 8 enzymatic reactions. This pathway takes place in the mitochondrial matrix, where the high concentration of enzymes and optimal ionic conditions facilitate efficient substrate turnover and energy conservation. Each turn of the citric acid cycle consumes one acetyl-CoA molecule and regenerates the 4-carbon acceptor molecule that allows the cycle to continue indefinitely. The pathway produces 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of GTP, which is readily converted to ATP. More importantly, the cycle generates carbon dioxide as a waste product, representing the complete oxidation of the carbon skeleton that entered as acetyl-CoA. The regulation of the citric acid cycle involves multiple control points that ensure coordination with cellular energy demands and metabolic status. Isocitrate dehydrogenase responds to the cellular energy charge through inhibition by ATP and stimulation by ADP, providing sensitive control over cycle activity. Alpha-ketoglutarate dehydrogenase is inhibited by its products succinyl-CoA and NADH, creating feedback regulation that prevents overproduction of reducing equivalents when the electron transport chain becomes saturated. The electron transport chain represents the culmination of cellular respiration, where the reducing power captured in NADH and H2 drives the synthesis of ATP through a process known as oxidative phosphorylation. This system consists of 4 large protein complexes embedded in the inner mitochondrial membrane, each containing multiple redox centers that facilitate the stepwise transfer of electrons from NADH and H2 to molecular oxygen. Complex I, also known as NADH dehydrogenase, accepts electrons from NADH and transfers them to ubiquinone, while pumping protons from the mitochondrial matrix to the intermembrane space. This complex contains more than 40 individual protein subunits and multiple prosthetic groups, including flavin mononucleotide and iron-sulfur clusters, that facilitate electron transfer while ensuring directional proton pumping. Complex III, the cytochrome BC1 complex, accepts electrons from ubiquinol and transfers them to cytochrome C while pumping additional protons across the membrane. The Q-cycle mechanism employed by this complex ensures efficient energy conservation by coupling electron transfer to proton translocation through a sophisticated mechanism that involves the sequential oxidation and reduction of ubiquinol at 2 distinct binding sites. Complex IV, cytochrome C oxidase, catalyzes the final step of electron transport by transferring electrons from cytochrome C to molecular oxygen, reducing it to water while pumping the final set of protons across the membrane. This reaction represents the reason why oxygen is essential for aerobic life, serving as the terminal electron acceptor that allows the complete oxidation of organic fuels. The chemiosmotic theory explains how the energy of electron transport drives ATP synthesis through the creation of an electrochemical gradient across the inner mitochondrial membrane. The protons pumped by the electron transport complexes accumulate in the intermembrane space, creating both a concentration gradient and an electrical potential that stores energy in a form analogous to a charged battery. ATP synthase harnesses this proton motive force to drive the synthesis of ATP from ADP and inorganic phosphate through a remarkable molecular motor mechanism. The enzyme consists of 2 main components: the F0 portion embedded in the membrane that allows proton flow, and the F1 portion that extends into the matrix and contains the catalytic sites for ATP synthesis. As protons flow through the F0 portion down their electrochemical gradient, they cause rotation of a central shaft that induces conformational changes in the F1 portion, driving ATP synthesis through a binding change mechanism. The efficiency of cellular respiration becomes apparent when considering the complete energy yield from glucose oxidation. Theoretical calculations suggest that 38 ATP molecules can be produced from 1 glucose molecule under optimal conditions, though actual yields are typically lower due to the metabolic costs of transporting molecules across membranes and maintaining cellular processes. Alternative fuels demonstrate the metabolic flexibility of cellular respiration pathways. Fatty acids undergo beta-oxidation to produce acetyl-CoA molecules that enter the citric acid cycle, generating even more ATP per carbon atom than glucose due to their highly reduced nature. Amino acids can be deaminated and converted to various intermediates of glycolysis, and the citric acid cycle, allowing proteins to serve as metabolic fuels during periods of carbohydrate restriction. Ketone bodies represent specialized fuel molecules produced by the liver during periods of prolonged fasting or carbohydrate restriction. Beta-hydroxybutyrate and aceto can cross the blood-brain barrier and provide alternative fuels for the brain when glucose availability is limited, demonstrating the remarkable adaptability of cellular energy metabolism. The intricate regulation of cellular respiration ensures that energy production matches cellular demands, while maintaining the metabolic flexibility necessary for survival under diverse nutritional and environmental conditions. This ancient pathway continues to power every cell in your body, converting the food you eat into the energy that sustains your thoughts, movements, and very existence. Photosynthesis stands as the most consequential biological process on Earth, fundamentally responsible for converting solar energy into the chemical energy that sustains virtually all life on our planet. This remarkable process transforms simple inorganic molecules into complex organic compounds, creating the foundation of food webs while simultaneously producing the oxygen that makes aerobic life possible. Without photosynthesis, Earth would remain a barren, lifeless world devoid of the rich biosphere that characterizes our planet today. The global significance of photosynthesis extends far beyond its role in individual plant cells. Every year, photosynthetic organisms convert approximately 100 billion tons of carbon dioxide into organic matter, representing an energy capture equivalent to burning 400 billion tons of coal. This massive energy conversion supports not only the plants themselves, but also the entire web of heterotrophic organisms that depend on photosynthetic products for their survival. The evolutionary origin of photosynthesis fundamentally altered Earth's atmosphere and climate, creating what scientists call the Great Oxidation Event, approximately 2.4 billion years ago. Before photosynthesis evolved, Earth's atmosphere contained virtually no free oxygen. The emergence of oxygen-producing cyanobacteria gradually transformed the atmospheric composition, enabling the evolution of aerobic respiration and complex multicellular life forms. Chloroplasts serve as the specialized organelles where photosynthesis occurs in eukaryotic plants and algae. These double membrane-bounded structures contain an intricate internal membrane system called thylakoids, which organize into stacked structures known as grana connected by unstacked stromal lamellae. This complex membrane architecture creates distinct compartments that facilitate the spatial separation of photosynthetic reactions, while maximizing the surface area available for light capture. The thylakoid membranes contain the molecular machinery responsible for capturing light energy and converting it into chemical energy. Photosystem complexes embedded within these membranes contain hundreds of chlorophyll molecules organized into antenna complexes that funnel absorbed light energy to reaction centers where photochemical conversion occurs. The precise arrangement of these pigment molecules ensures efficient energy transfer while preventing energy loss through fluorescence or heat dissipation. Chlorophyll molecules represent the primary light-harvesting pigments responsible for the green coloration of plants and the initial capture of solar energy. The molecular structure of chlorophyll, with its large porphyrin ring system and central magnesium atom, provides optimal absorption properties for capturing photons in the red and blue regions of the visible spectrum. Accessory pigments, including carotenoids and anthocyanins, extend the range of useful light wavelengths while providing protection against photodamage from excess light energy. The light reactions of photosynthesis occur within the thylakoid membranes and accomplish two essential functions: the production of ATP and the generation of NADPH, both of which provide the energy and reducing power necessary for carbon dioxide fixation. These reactions begin when photons strike photosystem II, exciting chlorophyll molecules and initiating electron transfer reactions that ultimately split water molecules and release oxygen as a byproduct. Photosystem II operates as a sophisticated molecular machine that extracts electrons from water through one of the most thermodynamically challenging reactions in biology. The oxygen-evolving complex contains a cluster of manganese and calcium ions that accumulate oxidizing equivalents through successive photochemical reactions until sufficient energy is available to oxidize water. This process requires the precise coordination of four photochemical events, explaining why oxygen evolution occurs in bursts rather than continuously. The electrons extracted from water by photosystem II travel through an electron transport chain that closely resembles the respiratory electron transport chain, but operates in reverse. Plastoquinone serves as a mobile electron carrier that transfers electrons from photosystem II to the cytochrome B6F complex, which pumps protons across the thylakoid membrane while transferring electrons to plastocyanin. This electron flow creates the proton gradient that drive ATP synthesis while preparing electrons for further energization by photosystem 1. Photosystem 1 captures additional light energy to boost electrons to even higher energy levels, enabling the reduction of NADP+ to NADPH. The reaction center of photosystem I contains a special chlorophyll molecule called P700 that becomes highly reducing when excited by light, allowing it to transfer electrons to ferredoxin and ultimately to NADP+, through the action of ferredoxin NADP+ reductase. The chemiosmotic mechanism of ATP synthesis in chloroplasts parallels the process that occurs in mitochondria, but operates in the opposite direction with respect to proton gradients. The thylakoid space becomes acidified as protons accumulate from water splitting and cytochrome B6F activity, while the stroma maintains a more alkaline pH. ATP synthase embedded in the thylakoid membrane harnesses this proton gradient to drive ATP synthesis, providing the energy currency needed for carbon dioxide fixation. The Calvin cycle represents the metabolic pathway responsible for incorporating atmospheric carbon dioxide into organic molecules through a process known as carbon fixation. This cyclical pathway occurs in the chloroplast stroma and requires the coordinated action of multiple enzymes to achieve the net synthesis of carbohydrate molecules from carbon dioxide. The cycle can be conceptually divided into 3 phases: carbon fixation, reduction, and regeneration of the starting compounds. Carbon fixation begins with the enzyme ribulose 1,15-bisphosphate carboxylase oxygenase, commonly known as Ribisco, which catalyzes the addition of carbon dioxide to ribulose 1,15-bisphosphate. This enzyme represents the most abundant protein on Earth, reflecting its central role in global carbon cycling. Despite its importance, Ribisco exhibits relatively slow catalytic rates and can mistakenly bind oxygen instead of carbon dioxide, leading to photorespiration, a process that reduces photosynthetic efficiency. The reduction phase of the Calvin cycle uses the ATP and NADPH generated by the light reactions to convert the initial carbon fixation products into glycolaldehyde 3-phosphate, a 3-carbon sugar that serves as the precursor for all other organic molecules synthesized by the plant. This phase demonstrates the direct coupling between light energy capture and carbon assimilation, illustrating how photosynthesis transforms solar energy into chemical bond energy. The regeneration phase ensures the continued operation of the Calvin cycle by using some of the glycolaldehyde 3-phosphate molecules to regenerate ribulose 1,15-bisphosphate, the carbon dioxide acceptor molecule. This complex series of reactions involves the rearrangement of carbon skeletons through various intermediates requiring precise stoichiometric relationships to maintain cycle function. Photosynthetic efficiency varies considerably depending on environmental conditions and the specific adaptations of different plant species. Under optimal conditions, plants can convert approximately 3 to 6% of incident solar energy into chemical energy, though this efficiency often decreases under stress conditions such as high temperatures, drought, or limited nutrient availability. C4 photosynthesis represents an evolutionary adaptation that concentrates carbon dioxide around Rubisco, reducing the enzyme's tendency to bind oxygen and improving photosynthetic efficiency under hot, dry conditions. Plants employing this strategy initially fix carbon dioxide into 4 carbon compounds in specialized mesophyll cells, then transport these compounds to bundle sheath cells where carbon dioxide is released in high concentrations around Rubisco. This spatial separation of initial carbon fixation from the Calvin cycle allows C4 plants to maintain high photosynthetic rates even when stomata close to conserve water. Crassolution acid metabolism, or CAM photosynthesis, provides another solution to the challenge of maintaining photosynthetic efficiency under water-limited conditions. CAM plants open their stomata at night to fix carbon dioxide into organic acids, then close their stomata during the day to conserve water while releasing the stored carbon dioxide for use in the Calvin cycle. This temporal separation of carbon dioxide uptake from light reactions allows desert plants to photosynthesize while minimizing water loss. The oxygen revolution triggered by the evolution of oxygenic photosynthesis represents one of the most dramatic environmental changes in Earth's history. The accumulation of oxygen in the atmosphere enabled the evolution of aerobic respiration, which provides much more ATP per glucose molecule than anaerobic metabolism. This increased energy availability facilitated the evolution of complex multicellular organisms and the diverse ecosystems that characterize modern Earth. demonstrating how photosynthesis continues to shape the conditions that support life on our planet. Metabolism encompasses the vast network of interconnected chemical reactions that sustain life, representing one of the most complex and elegant systems in biology. These pathways operate as integrated circuits where thousands of individual reactions coordinate to maintain cellular function, respond to environmental changes, and support the diverse energy demands of living organisms. Understanding metabolism requires appreciating both the individual components and the emergent properties that arise from their sophisticated organization. Metabolic pathways exhibit distinct organizational patterns that reflect their evolutionary origins and functional requirements. Linear pathways convert a starting substrate through a series of intermediates to a final product, with each step catalyzed by a specific enzyme. Branched pathways allow single substrates to be converted into multiple products, providing metabolic flexibility and enabling cells to produce diverse compounds from common precursors. Cyclic pathways regenerate their starting compounds while producing useful products or energy, creating self-sustaining metabolic engines that can operate continuously. The fundamental distinction between anabolic and catabolic processes provides a useful framework for understanding metabolic organization. Catabolic pathways break down complex molecules into simpler components, releasing energy that can be captured in high-energy bonds or used to drive other cellular processes. These degradative pathways typically involve oxidation reactions that remove electrons from substrates, generating reducing equivalents that can be used for energy production. Anabolic pathways construct complex molecules from simpler building blocks, requiring energy input to drive thermodynamically unfavorable reactions. These biosynthetic processes typically involve reduction reactions that add electrons to substrates, consuming the reducing power generated by catabolic pathways. The coupling of catabolism and anabolism through shared energy currencies like ATP and ADPH ensures that metabolic networks maintain overall energy balance while supporting both energy production and biosynthetic needs. Metabolic regulation operates through multiple layers of control that allow cells to respond rapidly to changing conditions while maintaining long-term metabolic stability. Allosteric regulation provides immediate control over enzyme activity through the binding of small molecule effectors that modulate enzyme conformation and catalytic efficiency. These regulatory molecules often represent the end products of metabolic pathways, creating feedback loops that prevent overproduction of specific compounds. Covalent modification of enzymes provides another rapid regulatory mechanism that can dramatically alter enzyme activity within seconds or minutes. Phosphorylation and dephosphorylation reactions, catalyzed by protein kinases and phosphatases respectively, allow hormonal signals to modulate enzyme activity throughout the cell. These modifications can activate or inhibit enzymes, alter their subcellular localization, or change their interactions with other proteins. Compartmentalization represents a sophisticated regulatory strategy that allows cells to maintain distinct metabolic environments within different subcellular locations. The separation of fatty acid synthesis in the cytoplasm from fatty acid oxidation in mitochondria prevents futile cycling while allowing both processes to operate when appropriate. Similarly, the compartmentalization of glucose metabolism between cytoplasm and mitochondria enables efficient energy production while maintaining metabolic flexibility. Hormonal control of metabolism provides long-term coordination of metabolic processes across multiple tissues and organ systems. Insulin promotes anabolic processes by stimulating glucose uptake, glycogen synthesis, and fatty acid synthesis while inhibiting gluconeogenesis and lipolysis. This hormonal signal effectively switches the body from a catabolic state focused on mobilizing stored fuels to an anabolic state focused on storing excess nutrients. Glucagon exerts opposing effects by promoting catabolic processes that mobilize stored fuels during periods of fasting. This hormone stimulates glycogenolysis and gluconeogenesis in the liver, while promoting lipolysis in adipose tissue, ensuring adequate fuel supplies for glucose-dependent tissues like the brain. The reciprocal relationship between insulin and glucagon creates a hormonal switch that coordinates whole-body metabolism with nutritional status. Cortisol provides a third layer of hormonal control that becomes particularly important during stress conditions. This steroid hormone promotes gluconeogenesis and protein catabolism while suppressing immune function and inflammation, redirecting metabolic resources toward immediate survival needs. The complex interactions between these and other hormones create sophisticated regulatory networks that maintain metabolic homeostasis under diverse physiological conditions. Metabolic diseases illustrate the critical importance of proper metabolic regulation and highlight the consequences of disrupted metabolic networks. Type 1 diabetes results from the autoimmune destruction of insulin-producing beta cells, eliminating the hormonal signal that promotes glucose uptake and storage. Without insulin, cells cannot efficiently utilize glucose, leading to hyperglycemia and the breakdown of protein and fat stores to provide alternative fuels. Type 2 diabetes involves insulin resistance, where target tissues become less responsive to insulin signaling despite normal or even elevated hormone levels. This condition often develops gradually in association with obesity and metabolic syndrome, representing a complex interplay between genetic predisposition, environmental factors, and lifestyle choices. The metabolic dysfunction in type 2 diabetes affects multiple organ systems and demonstrates how disrupted metabolic regulation can have far-reaching health consequences. Inborn errors of metabolism represent genetic disorders that result from deficiencies in specific enzymes or transport proteins. Phenylketonuria, caused by deficiency of phenylalanine hydroxylase, leads to accumulation of phenylalanine and its metabolites, causing intellectual disability if untreated. These disorders highlight the critical importance of individual enzymes in metabolic networks and demonstrate how single gene defects can disrupt entire metabolic pathways. Metabolic adaptation demonstrates the remarkable flexibility of metabolic networks in responding to environmental challenges. During prolonged exercise, muscle cells shift from primarily glucose metabolism to increased reliance on fatty acid oxidation, conserving glucose stores for tissues that cannot efficiently use alternative fuels. This metabolic flexibility requires coordinated changes in enzyme expression, hormone sensitivity, and substrate availability. Fasting triggers a coordinated metabolic response that maintains glucose homeostasis while transitioning the body to alternative fuel utilization. Initial responses involve glycogen breakdown and gluconeogenesis to maintain blood glucose levels. As fasting continues, increased lipolysis provides fatty acids for most tissues, while ketone body production by the liver provides alternative fuels for the brain and other glucose-dependent tissues. The emerging field of systems biology approaches metabolism as an integrated network where the properties of the whole system cannot be predicted from studying individual components in isolation. Metabolic flux analysis uses mathematical modeling to understand how metabolites flow through pathways under different conditions, revealing emergent properties that arise from network interactions. Metabolomics represents the comprehensive study of small molecule metabolites present in biological systems, providing snapshots of metabolic activity under specific conditions. This approach can identify biomarkers of disease, monitor responses to therapeutic interventions, and reveal previously unknown metabolic relationships. The integration of metabolomics with genomics and proteomics creates systems-level understanding of how genetic variation influences metabolic phenotypes. Personal metabolomics holds promise for precision medicine approaches that tailor therapeutic interventions to individual metabolic profiles. Understanding how genetic variation, environmental factors, and lifestyle choices interact to influence metabolic networks could enable personalized nutrition recommendations, optimized exercise prescriptions, and targeted therapeutic approaches for metabolic diseases. The study of metabolism continues to reveal new principles of biological organization and regulation while providing practical insights for improving human health. From the molecular mechanisms that control individual enzymes to the systems-level properties that emerge from network interactions, metabolic research demonstrates how the chemical reactions of life organize into the coherent patterns that sustain biological complexity. Cell signaling represents the fundamental communication system that allows individual cells to coordinate their activities and respond appropriately to environmental changes. This sophisticated molecular network enables single-celled organisms to detect and respond to their surroundings while allowing multicellular organisms to integrate the activities of trillions of cells into coherent physiological responses. Without effective cellular communication, complex life as we know it would be impossible. Signal transduction begins when cells detect specific chemical, physical, or electrical stimuli through specialized receptor proteins that recognize and bind to particular signaling molecules. This initial recognition event triggers a cascade of molecular interactions that amplify the original signal and ultimately produce appropriate cellular responses. The specificity of this process ensures that cells respond only to relevant signals while ignoring the countless other molecules present in their environment. The types of cellular signaling reflect the diverse communication needs of biological systems. Autocrine signaling allows cells to respond to signals they produce themselves, creating feedback loops that can either amplify or dampen cellular responses. This self-signaling plays important roles in cellular differentiation and immune responses, where cells must coordinate their own activities based on their internal state and external conditions. Paracrine signaling enables cells to communicate with nearby neighbors through the release of signaling molecules that diffuse over short distances. This local communication system coordinates tissue-level responses and ensures that neighboring cells can respond collectively to local changes in their environment. Growth factors and neurotransmitters often function as paracrine signals, allowing precise spatial control of cellular responses. Endocrine signaling provides long-distance communication through hormones that travel through the bloodstream to reach target cells throughout the body. This system enables the coordination of physiological responses across multiple organ systems, allowing the body to respond as an integrated unit to changes in nutritional status, stress levels, or reproductive state. The specificity of endocrine signaling depends on the presence of appropriate receptors on target cells rather than the physical proximity of signaling and responding cells. Neural signaling represents a specialized form of communication that combines electrical and chemical components to enable rapid, precise communication over long distances. Neurons use electrical signals to transmit information along their length and chemical neurotransmitters to communicate with other cells at synapses. This system provides the speed and precision necessary for complex behaviors and rapid responses to environmental changes. Receptor proteins serve as the molecular interfaces that detect signaling molecules and convert their presence into cellular responses. These proteins exhibit remarkable specificity, often distinguishing between closely related molecules through precise molecular recognition mechanisms. The binding of signaling molecules to receptors induces conformational changes that initiate downstream signaling events, demonstrating how molecular structure determines biological function. Membrane-bound receptors detect signaling molecules that cannot cross cell membranes, including most hormones, neurotransmitters, and growth factors. These receptors span cellular membranes and undergo conformational changes upon ligand binding that are transmitted across the membrane to initiate intercellular signaling cascades. The diversity of membrane receptor types reflects the variety of extracellular signals that cells must detect and respond to appropriately. G-protein coupled receptors represent the largest family of membrane receptors, mediating responses to light, odors, tastes, hormones, and neurotransmitters. These receptors interact with intercellular G-proteins that amplify receptor activation and initiate diverse signaling pathways. The modular organization of G-protein signaling allows different receptors to activate common downstream effectors, while maintaining specificity through distinct G-protein subtypes. Receptor tyrosine kinases respond to growth factors and hormones by undergoing dimerization and autophosphorylation upon ligand binding. The phosphorylated tyrosine residues serve as docking sites for intercellular signaling proteins, creating platforms for assembling signaling complexes that regulate cell proliferation, differentiation, and survival. Dysregulation of these receptors contributes to cancer development, highlighting their critical importance in cellular homeostasis. Intercellular receptors detect signaling molecules that can cross cell membranes, including steroid hormones, thyroid hormones, and some vitamins. These receptors often function as transcription factors that directly regulate gene expression in response to hormone binding. The ability of these receptors to control gene transcription allows them to produce long-lasting cellular responses that can fundamentally alter cell behavior and function. Second messenger systems amplify receptor activation and coordinate cellular responses through the production of small signaling molecules that rapidly diffuse throughout the cell. Cyclic adenosine monophosphate serves as a crucial second messenger that activates protein kinase A, leading to phosphorylation of numerous target proteins. This amplification mechanism allows a small number of activated receptors to influence thousands of proteins throughout the cell. Calcium ions function as versatile second messengers that regulate diverse cellular processes including muscle contraction, neurotransmitter release, and gene expression. The cellular machinery that controls calcium levels includes ion channels, pumps, and binding proteins that work together to create rapid, localized changes in calcium concentration. The temporal and spatial patterns of calcium signals encode information that cells use to generate appropriate responses. Lipid-derived second messengers demonstrate how membrane components can serve signaling functions beyond their structural roles. Diacylglycerol and inositol triphosphate, generated by phospholipase C activation, coordinate to regulate protein kinase C activity and calcium release from intracellular stores. These lipid messengers illustrate the integration of membrane metabolism with signal transduction pathways. Signal amplification represents one of the most remarkable features of cellular communication systems, allowing single receptor activation events to produce massive cellular responses. Enzymatic cascades achieve amplification through the sequential activation of multiple enzymes, with each activated enzyme catalyzing the activation of many downstream targets. This amplification enables cells to detect and respond to extremely low concentrations of signaling molecules. The phosphorylation cascades initiated by receptor activation demonstrate how protein modifications can rapidly alter enzyme activity and protein interactions throughout the cell. Protein kinases transfer phosphate groups from ATP to specific amino acid residues on target proteins, often dramatically changing their catalytic activity or binding properties. Protein phosphatase reverse these modifications, reaching dynamic systems where protein function is continuously regulated through phosphorylation state. Hormonal cascades illustrate how signal amplification extends beyond individual cells to coordinate responses across multiple tissues. The hypothalamic pituitary adrenal axis demonstrates this principle, where hypothalamic releasing hormones stimulate pituitary hormone release, which in turn stimulates adrenal hormone production. Each step in this cascade amplifies the original signal while providing additional opportunities for regulation and fine-tuning of the physiological response. Disrupted signaling pathways contribute to numerous disease states, highlighting the critical importance of proper cellular communication for health. Cancer often results from mutations that disrupt growth factor signaling, leading to uncontrolled cell proliferation and resistance to growth inhibitory signals. Understanding these signaling defects has led to targeted therapies that specifically interfere with the aberrant pathways driving tumor growth. Diabetes represents another example of signaling pathway disruption, where defective insulin signaling leads to impaired glucose uptake and metabolic dysfunction. The molecular basis of insulin resistance involves defects in receptor function, signal transduction, or downstream effector proteins that prevent normal cellular responses to insulin. These insights have guided the development of therapeutic approaches that target specific components of insulin signaling pathways. Pharmacology exploits cellular signaling mechanisms to develop therapeutic interventions that modulate specific pathways. Many drugs function as receptor agonists or antagonists, either mimicking or blocking the effects of natural signaling molecules. Understanding the molecular details of receptor function enables the design of drugs with improved specificity and reduced side effects, leading to more effective therapeutic interventions with fewer adverse consequences. Molecular genetics reveals how the information encoded in DNA sequences becomes the functional proteins that drive cellular processes, representing one of the most elegant information transfer systems in biology. This field illuminates the mechanisms by which genetic instructions are interpreted, regulated and executed to produce the remarkable diversity of cell types and functions that characterize complex organisms. Gene structure in eukaryotic organisms exhibits sophisticated organizational features that reflect the evolutionary complexity of these systems. Unlike the simple continuous coding sequences found in bacterial genes, eukaryotic genes contain both exons, which encode amino acid sequences, and introns, which are removed during RNA processing. This split gene architecture allows for alternative splicing patterns that can generate multiple protein variants from single genes, dramatically expanding the functional diversity of the genome. Promoter regions provide the regulatory sequences that determine when and where genes are transcribed into RNA. These DNA sequences, typically located upstream of the coding region, contain binding sites for RNA polymerase and various transcription factors that control gene expression. The core promoter includes essential elements like the Tartar box, which helps position RNA polymerase at the correct transcription start site, while additional regulatory elements fine-tune expression levels in response to cellular conditions. Enhancer and silencer sequences demonstrate how gene regulation can occur over vast genomic distances through DNA looping mechanisms that bring distant regulatory elements into contact with promoter regions. These regulatory sequences can be located thousands of base pairs away from the genes they control, yet still exert powerful effects on transcription through the formation of chromosomal loops that create functional regulatory complexes. Transcriptional regulation represents the primary control point for gene expression, determining which genes are active in different cell types and under various physiological conditions. Transcription factors serve as the key regulatory proteins that recognize specific DNA sequences and either promote or inhibit transcription initiation. These proteins often contain distinct domains for DNA binding and transcriptional activation or repression, allowing modular control of gene expression. The combinatorial nature of transcriptional regulation enables enormous regulatory complexity from a limited number of transcription factors. Different combinations of transcription factors binding to various regulatory elements can produce distinct expression patterns, allowing the same gene to be regulated differently in different cell types or developmental stages. This combinatorial control explains how relatively simple genomes can generate the complexity observed in multicellular organisms. Chromatin structure provides an additional layer of gene regulation through modifications to histones and DNA that affect the accessibility of genetic information. Histone modifications, including acetylation, methylation, and phosphorylation, create combinatorial codes that influence whether genes are in active or repressed states. These modifications can be inherited through cell divisions, providing a mechanism for maintaining cell-type-specific gene expression patterns. Post-transcriptional processing transforms the initial RNA transcript into mature mRNA through a series of carefully orchestrated modifications. The addition of a 5' cap structure protects the mRNA from degradation while facilitating ribosome binding during translation initiation. The 3' polyadenylation process adds a tail of adenine nucleotides that enhances mRNA stability and translation efficiency. RNA splicing removes introns from pre-mRNA transcripts through the coordinated action of small nuclear ribonucleoproteins that form the spliceosome complex. This process must occur with extraordinary precision, as errors in splicate recognition can lead to frame shifts or premature stop codons that produce non-functional proteins. Alternative splicing patterns allow single genes to produce multiple protein isoforms with different functions, greatly expanding the functional diversity of the proteome. microRNAs and other regulatory RNAs provide post-transcriptional control mechanisms that fine-tune gene expression by binding to complementary sequences in target mRNAs. These small regulatory RNAs can either promote mRNA degradation or inhibit translation, creating additional layers of control that allow rapid responses to changing cellular conditions without requiring changes in transcription rates. Translation control mechanisms regulate protein synthesis at multiple levels, from ribosome binding and initiation through elongation and termination. The 5' translated region of mRNAs contains regulatory sequences that influence ribosome binding efficiency and translation initiation rates. Internal ribosome entry sites allow some mRNAs to be translated even when normal cap-dependent translation is inhibited, providing mechanisms for maintaining essential protein synthesis under stress conditions. Post-translational modifications dramatically expand the functional diversity of proteins through chemical modifications that occur after translation. Phosphorylation represents the most common post-translational modification with protein kinases and phosphatases, creating dynamic systems where protein function is continuously regulated through phosphorylation state changes. These modifications can alter enzyme activity, protein localization, or protein-protein interactions. Ubiquitination marks proteins for degradation through the proteasome system, providing quality control mechanisms that remove misfolded or damaged proteins while allowing rapid changes in protein levels in response to cellular signals. The specificity of this system depends on E3 ligases that recognize specific protein targets and attach ubiquitin chains that serve as degradation signals. Epigenetic mechanisms create heritable changes in gene expression that do not involve alterations to DNA sequence itself. DNA methylation typically represses gene expression by interfering with transcription factor binding or recruiting proteins that establish repressive chromatin states. These methylation patterns can be maintained through DNA replication, allowing gene expression states to be inherited by daughter cells. Histone modifications create another form of epigenetic regulation through chemical modifications to the histone proteins around which DNA is wrapped. Histone acetylation generally promotes gene expression by relaxing chromatin structure, while histone methylation can either activate or repress genes depending on the specific residues modified. These modifications create stable but reversible changes in gene expression that can persist through multiple cell divisions. The role of epigenetics in development. demonstrates how identical genomes can generate diverse cell types through differential gene expression patterns. During development, cells progressively restrict their gene expression programs through epigenetic mechanisms that establish and maintain cell type-specific expression patterns. These mechanisms ensure that differentiated cells maintain their specialized functions while retaining the ability to respond to appropriate developmental or environmental signals. Cancer represents a disease fundamentally characterized by altered gene expression patterns that disrupt normal cellular growth control mechanisms. Oncogenes, when overexpressed or mutated, promote excessive cell proliferation and survival. Tumor suppressor genes normally prevent inappropriate cell division, and their inactivation through mutations or epigenetic silencing contributes to cancer development. The accumulation of multiple genetic and epigenetic alterations transforms normal cells into malignant tumors. The tumor suppressor protein P53 illustrates how single genes can coordinate complex cellular responses to DNA damage and other stress conditions. This protein functions as a transcription factor that activates genes involved in DNA repair, cell cycle arrest, and apoptosis. Mutations in P53 occur in approximately half of all human cancers, highlighting its critical role in maintaining genomic stability and preventing malignant transformation. Genetic engineering technologies have revolutionized both research and therapeutic applications by enabling precise manipulation of gene expression. Recombinant DNA techniques allow the insertion of genes into vectors that can be introduced into cells or organisms, enabling the production of therapeutic proteins or the creation of genetically modified organisms with desired traits. CRISPR-Cas systems provide unprecedented precision for genome editing by enabling targeted modifications to specific DNA sequences. This technology allows researchers to knock out genes, introduce specific mutations, or insert new genetic elements with remarkable efficiency and specificity. The therapeutic potential of CRISPR includes correcting disease-causing mutations and developing new approaches to treating genetic disorders. Gene therapy approaches aim to treat diseases by introducing functional genes into patients' cells to compensate for defective or missing genes. While early gene therapy trials face significant challenges, improved vector systems and delivery methods have led to successful treatments for several genetic disorders, demonstrating the therapeutic potential of genetic engineering approaches for treating previously incurable conditions. The biochemical basis of health and disease reveals how disruptions in fundamental molecular processes can cascade through biological systems, transforming normal cellular function into pathological states that compromise human well-being. Understanding these molecular mechanisms provides insight into both the remarkable resilience of biological systems and their vulnerabilities when critical biochemical pathways fail. Metabolic disorders represent a diverse group of conditions caused by defects in specific enzymes or transport proteins that disrupt normal biochemical pathways. Phenylketonuria illustrates how single enzyme deficiencies can have profound physiological consequences. This genetic disorder results from mutations in phenylalanine hydroxylase, the enzyme responsible for converting phenylalanine to tyrosine. Without functional enzyme activity, phenylalanine accumulates to toxic levels while tyrosine becomes deficient, leading to intellectual disability, behavioral problems, and other serious complications if left untreated. The biochemical logic underlying phenylketonuria treatment demonstrates how understanding metabolic pathways enables rational therapeutic approaches. Restricting dietary phenylalanine prevents substrate accumulation, while supplementing tyrosine addresses the deficiency created by the enzymatic block. This approach illustrates the fundamental principle that metabolic disorders often require lifelong dietary management to compensate for genetic defects. Galactosemia represents another inherited metabolic disorder where defective galactose metabolism leads to accumulation of toxic intermediates. Classical galactosemia results from galactose 1-phosphate uridyl transferase deficiency, causing galactose 1-phosphate to accumulate in tissues where it interferes with glycogen metabolism and protein synthesis. The resulting symptoms include liver dysfunction, cataracts, and intellectual disability, demonstrating how disrupted carbohydrate metabolism can affect multiple organ systems. Lysosomal storage diseases encompass a group of disorders where defective lysosomal enzymes lead to the progressive accumulation of undegraded substrates within cells. Tasostrosase exemplifies this category, involving hexosaminidase A deficiency that prevents the breakdown of GM2 gangliosides in nerve cells. The progressive accumulation of these lipids destroys neuronal function, leading to developmental regression and early death. These disorders highlight the critical importance of cellular recycling mechanisms for maintaining normal cell function. Protein misfolding diseases demonstrate how the three-dimensional structure of proteins determines their biological function and how structural abnormalities can lead to devastating pathological processes. Alzheimer's disease involves the aggregation of amyloid beta peptides and tau proteins into fibrillar structures that accumulate in brain tissue. These protein aggregates disrupt normal neuronal function through multiple mechanisms, including interference with cellular transport, oxidative stress generation, and activation of inflammatory responses. The prion diseases represent the most extreme example of protein misfolding pathology, where abnormally folded proteins can induce normal proteins to adopt the misfolded conformation. The prion protein can exist in both normal cellular forms and pathological scrapie forms that differ only in their three-dimensional structure. The scrapie form acts as a template that converts normal prion proteins to the abnormal conformation, creating a self-propagating process that leads to progressive neurodegeneration. Parkinson's disease involves the misfolding and aggregation of alpha-synuclein protein in dopamine-producing neurons of the substantia nigra. These protein aggregates, called Lewy bodies, contribute to neuronal dysfunction and death, leading to the movement disorders characteristic of Parkinson's disease. The progressive loss of dopaminergic neurons disrupts the neural circuits controlling movement, demonstrating how protein misfolding can target specific cell populations. Enzyme deficiencies illustrate how the loss of specific catalytic activities can disrupt entire metabolic networks. Glucose 6-phosphate dehydrogenase deficiency affects the pentose phosphate pathway, reducing the cell's ability to generate NADPH and maintain antioxidant defenses. This deficiency particularly affects red blood cells, which lack alternative mechanisms for generating reducing power, leading to hemolytic anemia when patients are exposed to oxidative stress from certain drugs or foods. The biochemical consequences of enzyme deficiencies often extend far beyond the immediate metabolic pathway involved. Glycogen storage diseases result from defects in enzymes involved in glycogen metabolism, leading to abnormal glycogen accumulation in various tissues. Vangerka disease, caused by glucose 6-phosphatase deficiency, prevents glucose release from liver glycogen, leading to severe hypoglycemia and metabolic acidosis. These systemic effects demonstrate how localized enzyme defects can disrupt whole-body glucose homeostasis. Oxidative stress represents a common pathway through which various disease processes damage cellular components and disrupt normal function. This condition arises when the production of reactive oxygen species exceeds the capacity of antioxidant defense systems to neutralize them. Reactive oxygen species can damage proteins, lipids, and nucleic acids, leading to cellular dysfunction and contributing to aging, cancer, and degenerative diseases. Antioxidant defense systems protect cells against oxidative damage through multiple complementary mechanisms. Enzymatic antioxidants like superoxide dismutase, catalase, and glutathione peroxidase directly neutralize reactive oxygen species through specific chemical reactions. Non-enzymatic antioxidants including vitamin C, vitamin E, and glutathione provide additional protection by scavenging free radicals and preventing chain reactions that amplify oxidative damage. The balance between oxidative stress and antioxidant defenses influences aging processes and disease susceptibility. Mitochondrial dysfunction contributes to oxidative stress by increasing reactive oxygen species production, while decreasing ATP synthesis efficiency. This creates a vicious cycle where oxidative damage impairs mitochondrial function, leading to further oxidative stress and cellular dysfunction. Inflammation represents the body's response to tissue damage, infection, or other threats, involving complex biochemical cascades that coordinate immune responses while promoting tissue repair. The inflammatory response involves multiple classes of signaling molecules, including cytokines, chemokines, and lipid mediators that coordinate the recruitment and activation of immune cells at sites of tissue damage. The biochemical mediators of inflammation include prostaglandins and leukotrienes derived from arachidonic acid metabolism. These lipid signaling molecules promote vasodilation, increase vascular permeability, and attract immune cells to sites of inflammation. While these responses are essential for fighting infections and promoting healing, chronic inflammation can contribute to numerous diseases including cardiovascular disease, diabetes, and cancer. The resolution of inflammation requires active biochemical processes that terminate inflammatory responses and promote tissue repair. Specialized pro-resolving mediators derived from omega-3 fatty acids help resolve inflammation by promoting the clearance of dead cells and the return of tissue homeostasis. Defects in inflammation resolution can lead to chronic inflammatory diseases that cause progressive tissue damage. Aging involves the progressive accumulation of molecular damage that gradually impairs cellular function and increases disease susceptibility. Telomeres, the protective DNA protein structures at chromosome ends, shorten with each cell division and eventually triggers cellular senescence when they become critically short. This process limits the replicative capacity of cells and contributes to aging-related changes in tissue function. Cellular senescence involves the permanent growth arrest of cells that have experienced excessive stress or damage. Senescent cells secrete inflammatory factors that can promote tissue dysfunction and contribute to age-related diseases. The accumulation of senescent cells with age represents one mechanism by which cellular damage translates into organismal aging. Nutritional biochemistry reveals how dietary components influence cellular function and disease risk through their roles as enzyme cofactors, antioxidants, and signaling molecules. Vitamins function as essential cofactors for numerous enzymatic reactions, and vitamin deficiencies can disrupt multiple metabolic pathways simultaneously. Vitamin deficiency diseases like scurvy and pellagra demonstrate how nutritional inadequacies can cause severe systemic dysfunction. Mineral nutrients play critical roles in enzyme function, protein structure, and cellular signaling. Iron deficiency affects oxygen transport and energy metabolism through its roles in hemoglobin and cytochromes. Zinc deficiency impairs immune function and wound healing through its roles in numerous enzymes and transcription factors. These examples illustrate how nutritional status directly influences biochemical processes that determine health outcomes. Pharmacokinetics describes how the body processes medications through absorption, distribution, metabolism, and excretion. Understanding these processes is essential for optimizing drug therapy and minimizing adverse effects. Drug metabolism occurs primarily in the liver through cytochrome P450 enzymes that chemically modify drugs to facilitate their elimination. Genetic variations in these enzymes can dramatically affect individual responses to medications, highlighting the importance of personalized medicine approaches. The biochemical basis of drug action involves specific interactions between medications and their molecular targets. Many drugs function by inhibiting specific enzymes or blocking receptor proteins, demonstrating how understanding biochemical mechanisms enables rational drug design. The development of targeted cancer therapies illustrates how detailed knowledge of disease mechanisms can lead to highly specific therapeutic interventions that minimize side effects while maximizing efficacy. Biotechnology represents the convergence of biological understanding and technological innovation, transforming our comprehension of biochemical processes into practical applications that address fundamental challenges in medicine, agriculture, and environmental sustainability. This field demonstrates how detailed knowledge of molecular mechanisms enables the development of powerful tools for manipulating biological systems to achieve desired outcomes. Recombinant DNA technology forms the foundation of modern biotechnology by enabling the precise manipulation of genetic material to produce desired proteins or modify cellular functions. This approach involves isolating genes of interest, inserting them into suitable vectors, and introducing these constructs into host cells that can express the encoded proteins. The development of restriction enzymes that cut DNA at specific sequences and ligases that rejoin DNA fragments provided the molecular tools necessary for genetic engineering. The production of human insulin through recombinant DNA technology illustrates the transformative potential of biotechnology for medical applications. Before genetic engineering became available, diabetic patients relied on insulin extracted from animal pancreases, which sometimes caused allergic reactions, and was subject to supply limitations. The insertion of human insulin genes into bacterial cells enabled the large-scale production of human insulin that is identical to the hormone produced by pancreatic beta cells. The process of producing recombinant proteins involves multiple steps that require precise control of cellular conditions. Host cells must be engineered to express foreign genes efficiently while maintaining proper protein folding and post-translational modifications. Purification systems must separate the desired protein from thousands of other cellular proteins while maintaining biological activity. These technical challenges have driven innovations in fermentation technology, protein purification methods, and quality control systems. Gene therapy represents one of the most promising applications of biotechnology for treating genetic diseases, by introducing functional genes into patient cells to compensate for defective or missing genetic information. Early gene therapy approaches face significant challenges, including immune responses to viral vectors, difficulty targeting specific cell types, and problems with sustained gene expression. However, Recent advances in vector design and delivery methods have led to successful treatments for several genetic disorders. The treatment of severe combined immunodeficiency through gene therapy demonstrates how genetic engineering can address previously incurable conditions. This disorder results from mutations that prevent normal immune system development, leaving patients vulnerable to life-threatening infections. Gene therapy approaches introduce functional copies of the defective genes into patient stem cells, enabling normal immune system development and providing long-term protection against infections. CRISPR-Cas systems have revolutionized genome editing by providing unprecedented precision and efficiency for making targeted modifications to DNA sequences. This technology, originally discovered as a bacterial immune system that defends against viral infections, has been adapted to enable precise editing of genes in virtually any organism. The programmable nature of CRISPR allows researchers to target specific DNA sequences by designing guide RNAs that direct the Cas proteins to desired genomic locations. The therapeutic potential of CRISPR technology extends beyond simple gene correction to include sophisticated approaches for treating complex diseases. Base editing techniques enable the conversion of specific nucleotides without creating double-strand breaks, allowing precise correction of point mutations that cause genetic diseases. Prime editing provides even greater precision by enabling insertions, deletions, and replacements of DNA sequences with minimal off-target effects. Clinical applications of CRISPR have shown remarkable success in treating sickle cell disease and beta thalassemia by editing patients' hematopoietic stem cells to reactivate fetal hemoglobin production. This approach bypasses the defective adult hemoglobin genes, while restoring normal oxygen-carrying capacity through increased fetal hemoglobin expression. The success of these trials demonstrates the therapeutic potential of genome editing for treating genetic blood disorders. Synthetic biology represents an emerging field that applies engineering principles to biological systems, enabling the design and construction of biological devices, systems, and organisms with novel functions. This approach treats biological components as standardized parts that can be assembled into circuits with predictable behaviors. Synthetic biology applications range from microorganisms, engineered to produce valuable chemicals, to biological sensors that detect environmental pollutants. The engineering of metabolic pathways in microorganisms demonstrates how synthetic biology can create biological factories for producing valuable compounds. Researchers have engineered bacteria and yeast to produce pharmaceuticals, biofuels, and industrial chemicals through the introduction of novel metabolic pathways. These approaches can reduce production costs while minimizing environmental impact compared to traditional chemical synthesis methods. Biomarkers represent molecular indicators of biological processes, disease states, or responses to therapeutic interventions. The identification and validation of biomarkers requires sophisticated analytical techniques that can detect and quantify specific molecules in biological samples. Advances in mass spectrometry, proteomics, and genomics have enabled the discovery of biomarkers that can diagnose diseases earlier, predict treatment responses, and monitor therapeutic efficacy. Molecular diagnostics utilizes biomarkers to provide precise information about disease states and treatment options. Liquid biopsies analyze circulating tumor DNA to detect cancer mutations and monitor treatment responses without requiring invasive tissue sampling. Pharmacogenomic testing identifies genetic variants that influence drug metabolism and response, enabling personalized medication selection and dosing. Personalized medicine represents the ultimate application of biotechnology for tailoring medical treatments to individual patients based on their genetic makeup, biomarker profiles, and other molecular characteristics. This approach recognizes that genetic variation among individuals influences disease susceptibility, treatment responses, and adverse drug reactions. Personalized medicine aims to optimize therapeutic outcomes while minimizing side effects through precision targeting of interventions. The development of targeted cancer therapies illustrates how personalized medicine can transform treatment approaches. Rather than using broad-acting chemotherapy agents that affect all rapidly dividing cells, targeted therapies specifically inhibit molecular pathways that drive tumor growth. Trastuzumab targets the HER2 receptor in breast cancers that overexpress this protein, while imatinib inhibits the BCR-ABL kinase in chronic myeloid leukemia. These approaches achieve better outcomes with fewer side effects compared to traditional chemotherapy. Biofuel production through biotechnology offers sustainable alternatives to fossil fuels by harnessing biological processes for energy generation. Microorganisms can be engineered to convert biomass into ethanol, biodiesel or other fuels through optimized metabolic pathways. Algae represent particularly promising biofuel sources because they can produce large quantities of lipids while growing rapidly on non-arable land using minimal fresh water. The development of cellulosic biofuels involves engineering microorganisms to break down cellulose from agricultural waste into fermentable sugars. This approach could provide renewable fuels without competing with food production for agricultural land. Advances in enzyme engineering and metabolic pathway optimization continue to improve the efficiency and economics of biofuel production processes. Environmental biotechnology applies biological systems to address pollution and environmental degradation through bioremediation approaches that use microorganisms to degrade contaminants. Engineered bacteria can break down oil spills, heavy metals, and toxic chemicals into harmless products. These biological approaches often provide more environmentally friendly alternatives to traditional remediation methods that may create additional pollution. The future of biotechnology promises even more sophisticated applications that will further transform medicine, agriculture, and environmental management. Advances in artificial intelligence and machine learning are accelerating the discovery of new therapeutic targets and the optimization of biological systems. The integration of multiple biotechnology approaches will enable solutions to complex challenges that require coordinated interventions at molecular, cellular, and organismal levels. Emerging technologies like organ-on-chip systems provide new platforms for drug testing and disease modeling that could reduce reliance on animal testing while providing more accurate predictions of human responses. These systems recreate the functions of human organs using engineered tissues maintained in microfluidic devices, enabling researchers to study disease mechanisms and test therapeutic interventions in controlled human-relevant models. The continued development of biotechnology depends on maintaining public trust through responsible research practices, transparent communication about risks and benefits, and careful consideration of ethical implications. As biotechnology capabilities continue to expand, society must thoughtfully evaluate how these powerful tools should be used to ensure they benefit humanity, while minimizing potential risks and unintended consequences. The molecular processes we've explored represent more than just chemical reactions. They embody the fundamental principles that govern all living systems, from the simplest bacterial cell to the most complex human organ. The same biochemical themes repeat: energy transformation, information storage and transfer, structural organization, and responsive adaptation to environmental change. Perhaps most remarkably, these processes occur with extraordinary precision and efficiency, largely outside our conscious awareness. Every second, millions of enzymatic reactions proceed in each of your cells, maintaining the delicate balance between order and chaos that defines life itself. The ATP that powers your thoughts, the proteins that maintain your cellular architecture, the enzymes that repair molecular damage, all operate through principles we can now understand at the atomic level. This knowledge transforms how we approach health, disease, and human potential. Understanding biochemistry allows us to appreciate why certain foods nourish us, how exercise strengthens our bodies at the molecular level, and why sleep and stress management are biochemical necessities rather than lifestyle choices. It reveals the mechanistic basis for medical treatments and points toward future therapies that work with, rather than against, our evolved biochemical systems. Yet, for all our advancing knowledge, biochemistry continues to humble us with its complexity and elegance. Each answered question reveals new mysteries. Each solved pathway illuminates previously unknown connections. We are, quite literally, still discovering ourselves at the molecular level. In closing your eyes tonight, consider that you carry within yourself a biochemical universe of stunning sophistication, one that connects you to every living thing that has ever existed, and that continues to write the molecular story of life itself, one reaction at a time.